scientist is a bit surprised when the well-blinded me was.
Thank you. Thank you very much. Thank you for being here this afternoon. It was an interesting question put to you earlier when this began to see how many of you had been at the previous 4 events. Some of you, fortunately or unfortunately, have endured this discussion by me for 4 times. And you probably could get up here and do it about as well as I can. Others of you are hearing this for the first time, and it's a little bit of a challenge to know where to begin. I don't want to keep saying the same thing over and over. I want to tell you what's new. And so what I've done is put a really brief introduction of about 3 or 4 slides. If you're starting from ground zero, it's a little tough to jump in. But the rest of the time, I'm talking about things that are new. So that's what I'm going to attempt to do. I want to begin by introducing a term I haven't used with you before, but you need to start using this. The term is nutrigenomics. And what does that term mean? It's how dietary components, how things we eat, things we ingest, generally refers to food products, how they interact with the genome, that is the DNA that encodes all 25,000 of your genes to regulate gene expression. So those genes are the blueprints for everything that you are, and you can be put together by about 25,000 little unique machines. Some of them are unique to you. Nobody else has quite the same sequence. Others we all have in common, and that's the blueprint that makes you who you are. So how things we eat can affect the expression of those blueprints, how the rate at which they're converted into the enzymes that they encode, the little machines that do the things you have to do. This word you all know already, but what is it? What is Protandim? And the definition actually has changed a bit in the last 5 years. In the beginning, what we thought Protandim would be was an antioxidant supplement. Not a conventional one, but a different way of doing it. And that's no longer an adequate description. So this is what I want you to learn and want you to use as you describe Protandim. Protandim is a NRF2 activator in the nutrigenomics arena, all right? You may not have any idea what I just said, but you have a job, you have an obligation to learn what this means. And I'm going to tell you in the rest of this presentation what some of those words mean. NRF2, NRF2, it's an abbreviation for something you don't even want to hear, but it's a long word. NRF2 is one of the key proteins in your body, in every cell in your body, that has the function of regulating your genome. Yeah, I said you have 25,000 genes that are expressed. NRF2 regulates about 2% of those, about 500 genes, some of which are turned up, some of which are turned down. You can think of every gene as having a dimmer switch, so just like the lights in your home, and you can turn them up gradually or down. It's not just on and off, it's degrees. And NRF2 helps your body balance the expression, the regulation of all those 25,000 blueprints. How many copies do we need of this machine encoded by this blueprint? And NRF2 regulates that group, as I said, of 500 genes. They have something in common. They are termed generally survival genes, and they fall into 3 broad categories. Antioxidant enzymes, you've heard about superoxide dismutase catalase that protect you from oxidative stress. But another significant number of those genes are anti-inflammatory genes. They fight the process of inflammation in your body. And another group of them are anti-fibrotic genes. Means they fight the process of scar tissue formation. You might think scar formation is a good thing, and if you cut your skin, it is a good thing. It heals quickly and keeps you from getting infected, and it keeps you from losing blood, all good things. But in internal organs, in your lungs, scar tissue is deadly. It's called pulmonary fibrosis. It's untreatable, and it's fatal. If scar tissue occurs in your heart, it's called heart failure. It means your pump actually fails, and you will die of heart failure. So taken together, these things protect cells in your body when they're in stressful situations. It may be due to trauma, may be due to aging, it may be due to disease processes. But that group of genes all have the same kind of dimmer switch, and NRF2 is the protein that can reach those dimmer switches and turn them up or down. So it's responsible for that. It's been called the master regulator of antioxidant gene expression. So how does that work in a cell? This large green oval represents a human cell. And that little purple gizmo in the upper right, labeled a NRF2 activator, is one of the 5 active ingredients in Protandim. And so as that NRF activator approaches a human cell, it binds at a specific site. It's called a receptor on the cell surface. The analogy I'm going to use is if this big cell is your home, that NRF2 activator just stepped on the front porch, and it did something. It pushed your doorbell. It rings, causes a reaction inside the house, in this case, inside the cell. What is that reaction? Well, if it were the doorbell, you may be sitting on the sofa watching TV, and suddenly you get up, you do something, you have a response. Inside this cell, the same thing happens. When NRF2 rings the doorbell, as it were, an enzyme called a kinase, what it does is not terribly important, but it's one of these 25,000 gene products, and it's doing its job here to realize that NRF2 needs to be activated. So this NRF2 activator, by activating the kinase, brings about a chemical reaction, and there you see a little round red protein labeled NRF2. That's our signal molecule, messenger molecule. And it's being held inside the cell, but outside the nucleus by another protein here labeled KEAP1. Because this kinase physically changes the NRF2, and you can see there it's put a little yellow phosphate group on it, change in structure. And that change brings about a release of NRF2 from that blue protein that's been holding it out in the green part of the cell, allows it to diffuse into the nucleus where all those genes are housed. And once in there, it has access to all those dimmer switches, the 500 that control the NRF2 regulated genes. And so some of them will go up, some of them will go down, because NRF2 has entered the vault where the blueprints are kept. And this may seem like hand-waving, it may seem like a fantasy. Every step of this is true. It's simplified a little bit, as you might guess. And if I showed you the real science, you probably would have trouble following it. But that's what happens. And the analogy is not much different than if somebody rings your doorbell, you get up, answer the door, it tells you, go to the kitchen, and you enter that room of the house. And once in there, you can turn on switches. That's what NRF2 does. And when those switches are turned on, these 3 groups of genes that I just talked to you about are up and down regulated, depending on what they do. So antioxidant enzymes are expressed, more antioxidant enzyme protection. Antifibrotic genes are expressed, anti-inflammatory genes are expressed. And some genes are turned down, the ones that are pro-fibrotic and pro-inflammatory, because they're switches that cause things to happen and cause things not to happen. And the result is that cell is now protected from stress. And that was the review, and it was a fast course if you haven't encountered this before. But come back and you'll hear me elaborate on it from time to time. What I want to do now is talk about 3 new topics. And the first one, if you were at the last Elite Academy, unfortunately I gave Josh the wrong set of slides, and I didn't have the slides for the last topic that I wanted to talk about, and it was a topic that came up as a result of talking to you, because I was hearing things about, for example, people being able to stop smoking once they started taking Protandim. I started searching the literature, and it turned out there's a real story there, and dots were connected. And I want to tell you in a little more detail than I was able to last time what that's about. It's an idea at this point. It hasn't been validated. You've given me a lot of anecdotal feedback, and I'm more convinced than ever that this will pan out. But let me tell you how it works. Can Protandim help in the management of addictive behavior? And I'm going to talk about a couple of really bad addictive behaviors. Cocaine, which I hope affects nobody in here, and nicotine, which probably has affected some of you in here. And the amazing thing for science is just in the last few years, we've learned how these things work in the brain and what they have in common. And I, for one, was surprised that the mechanism by which cocaine causes addiction is not fundamentally different from the mechanism that nicotine causes addiction. And there have been other papers just published in the last couple of months that extend this, perhaps, to things like food addiction. And if you think about it, they're really not that different. The longing sensation you have for food is a survival value, because if you forgot about it, you didn't get hungry, you wouldn't eat, and eventually you'd lay down and die. So your brain has evolved to keep reminding you of what you need to do and cause you to get hungry and eat, keep your energy levels up. What this story is about is how some chemical substances, some molecules, have learned how to take control of that remarkable system to create their own sense of longing that needs to be satisfied, and paradoxically, to satisfy it acutely. Now, let me show you how this is now understood to work. This is a brain sliced front to back. And you see right there in the middle, there's a red region labeled N accumbens, nucleus accumbens. That has been designated by scientists who study these things as the pleasure center of the brain. And so it controls a lot of what you do, controls things like hunger, things like desire, and satiation from those feelings. So there's a specific part of the brain where this is handled. Brain cells communicate with one another by releasing chemicals so that one neuron can release these chemicals when it's at the region of that cell that's very close to the next neuron. Neurons are like short segments of wire, if you will. So you need to get a signal from one conductor to the next. And this is called firing of nerve cells, firing of neurons. And it's chemically induced. So one cell releases a neurotransmitter. That signal is picked up. It's like a speaker and a microphone. Picked up by the next cell, which can send that signal down its length. And some of the neurons in your body are up to 3 feet long, not in your brain, but in your periphery. So they're conductors signaling each other by this method. One of those neurotransmitters that's very important in the brain, there are a lot of them, serotonin, dopamine, you may have heard about. One of them is glutamate, and you've probably heard about glutamate too, in the form of MSG. Do you know what MSG is? It's responsible for Chinese restaurant syndrome. It is, MS is monosodium glutamate. MSG is monosodium glutamate. So you're actually using as a seasoning. It tastes good. And in most of your body, it has no effect. But in your brain, it actually has a neurotransmitter effect. And you can feel the effects of it. You get flushed. You feel hot. Your capillaries may dilate. So it's actually affecting neurotransmission in the brain. in this particular region of the brain that I'm talking about, the nucleus accumbens, that feeling of satisfaction is due to maintaining a basal level of glutamate. And again, this is a common metabolite. The rest of your body's making it. The brain is putting it to a particular use. And so it's been found just recently that cocaine blunts the activity of this region of the brain to release glutamate by downregulating a gene that enables glutamate to get into the cell. And that gene is called cysteine glutamate transporter, sometimes antiporter. And I'll tell you a little more about how that works. Here's a cell, and the yellow protein you see on the membrane of that cell, labeled XCT, is the cysteine glutamate exchange protein. How does that work? It works exactly like a revolving door on an office building. And what it does is it lets cysteine, one molecule, that's an amino acid, into the cell, but it only lets it in if there's a glutamate inside coming out the revolving door at the same time. That's why it's called an exchanger or an antiporter. And we have a lot of proteins on the cell surface that do exactly that. They let things in, let things out. Some of them actively pump things in and pump things out. Others are passive. This one will let cysteine in only if glutamate comes out. Now, the cysteine is very important, and I had questions this morning about glutathione. Glutathione is an important antioxidant. It's one of the antioxidants dramatically affected by Protandim because among the 500 genes Protandim regulates are the ones seen here in blue, glutathione synthase, that enables the cell to make its own glutathione. So if that enzyme goes up and more cysteine comes into the cell, you can make more glutathione. That cell is protected from oxidative stress. What we're looking at here is the reverse part of that revolving door. Every time a cysteine comes in, which enables the cell to make glutathione, a good thing, this brain cell pumps out one glutamate. And that glutamate is used to maintain a level of satisfaction in the brain. What we found by reading the literature was that cocaine blocks that revolving door. It downregulates that gene. And so if this building, this cell, let's say had 100 of these revolving doors letting cysteine and glutamate out, after taking cocaine for a month or two, you might only have 25% of those revolving doors still functioning. Okay, that's a concern. How does that manifest? And before we go there, let me say that the surprising part, nicotine, exactly the same thing. Nicotine affects the number of revolving doors the same way. So it decreases your brain's ability to make glutathione and decreases the brain's ability to secrete this important neurotransmitter. So chronically, either nicotine or cocaine, and I'm talking about nicotine because that's what's been published in more detail now, drops the basal level, the baseline level of glutamate, and what that does is it creates a sense of longing. So you see on the left side, normally you have a fairly high glutamate level, and on the right side, that's a situation that's neither longing nor satisfaction. It's just kind of a normal place to be. And if you take hits of nicotine to this region of the brain, and that's shown at the bottom, along the bottom of this axis, and you get repeated exposure to nicotine or cocaine, what happens is the glutamate level starts to drop. This is the baseline level. And that creates a sense of longing, a hunger for something. You need something. And here's the diabolical part. That's, let's say if we look daily or weekly after you start, say, smoking cigarettes, this would happen chronically over that week's time period. If you look at what happens in the short term, here, whoops. Let me back up one. Here. The red line shows you that the same exposure to nicotine or cocaine has a short burst of actually releasing glutamate. Okay, so it's doing two things: chronically starving those cells for glutamate, creating a longing, and acutely, meaning in the case of nicotine, maybe for only 15 minutes, causing those neurotransmitters to be released at the neuron junctions by a different mechanism than the one I just described. So it's affecting glutamate two different ways. It's blocking it in the basal level, and it's stimulating it for a short time. And you can see how diabolical that turns out to be, because taking repeated hits of these substances creates a desire, and the same substance temporarily relieves that desire. So as you move down this curve week after week, your longing increases, and you're dependent upon that spike to bring you back up to a level of satisfaction, right? If you were designing a diabolical system, it couldn't get any better than that. Okay, what about this turnstile protein? We have, in the past year, looked at the effect of Protandim on every gene in the human body, all 25,000. What I'm showing you here is a section of a spreadsheet that looks at about 25 of those genes. And these are the ones that are most affected by Protandim. And so there would be 1,000 more pages like this, and I have all of those, and I've spent hours looking through them. Now, as though I didn't have anything better to do with. And what you see here is, about 10 lines down is this cysteine glutamate transporter. It's one of the genes most affected by Protandim. And here we see that this, I think, is in brain cells. We've done it in vascular cells as well, at 2 different concentrations of Protandim each. And that 6.49 is at a high concentration of Protandim. And what that means is it increased the number of those turnstile proteins 6.5-fold, 649% more in the presence of Protandim. And this is it in bar graph form, showing you first what nicotine does. And so we're starting with normal, healthy cells, normalizing their level of this turnstile protein that's causing this problem, before and after nicotine. And what you can see is the nicotine really represses the expression of that protein. So the cells don't have these revolving doors. They can't get cysteine in to make glutathione. They can't get glutamate out to satisfy your brain. What does Protandim do to the same protein? It upregulates it. In this case, at a low dose of Protandim in endothelial cells, vascular cells, 250% in the other direction. All right? So this, again, this is not a human clinical trial. This is not even an animal study. These are putting together 2 dots and drawing a line between them, because we think we know how this system works and what will happen. And so the question raised by all of this is, can Protandim help break addictive behavior? And elevating GSH has been shown, on the one hand, to be efficacious in humans trying to break addictive behavior by increasing, or by methods that cause increased glutathione in this region of the brain. And they can break behavior to both cocaine and nicotine. We know that Protandim elevates GSH levels, and it's in part by increasing that turnstile protein. And what we found is that, or what has been found by others, is that that turnstile protein is also important in preventing this longing sensation from developing in the brain. So the turnstiles are absolutely essential. And then finally, anecdotal evidence, much of which I gleaned from you at the last Elite Academy, and a lot since then, suggests that indeed Protandim might be a useful adjunct, and that clinical trials to really prove this are probably an appropriate question for us to ask. So that's where we are. Again, many of you who have connections to smoking cessation clinics and so forth have contacted me. I think we're well on our way to providing an answer for this, and what an answer that would be. I mean, this is a real need in society at a number of levels, and there is no good way. Some of you probably stopped smoking in your lives, and it's not an easy thing. It's a tough thing, and I have total sympathy with those who are trying to do that. All right, that's number one. Number two, I want to inform you of a new human clinical trial. This is about the use of Protandim in the metabolic syndrome. And I'll tell you more what metabolic syndrome is if you're not familiar with it. The principal investigator on this project is one of my colleagues at the University of Colorado in Denver, Robert Eckel. And you probably don't recognize his name, but let me tell you who he is. He's one of America's foremost endocrinologists studying heart disease and metabolic syndrome. In 2005, Bob Eckel was the president of the American Heart Association. All right, so this is not chopped liver. This is a major study by a major player in the field, and it's just beginning. What is metabolic syndrome? It's a combination of disorders that increase the risk of developing cardiovascular disease, heart disease. It affects probably at least 20%, some estimate 25% of the American population. And it increases with age. And there are a number of, every organization like the American Heart Association and many others around the world have their criteria for diagnosing or predicting when you have metabolic syndrome. And usually there are these four conditions at the bottom: central obesity, elevated triglycerides, good HDL, low measure of the good form of LDL, of cholesterol, hypertension, high blood pressure, and fasting plasma glucose that's higher than it should be. That is hyperglycemia, which is an indication of developing diabetes. And usually 2 of those 4 is enough to say you're at risk for cardiovascular disease, you have metabolic syndrome. If you're not, this sounds like a Jeff Foxworthy joke, but if you're not sure if you have metabolic syndrome or not, you can look in the mirror. This is what it looks like. It's not a pretty picture, and it's called central obesity for the very obvious reason. It's what physicians describe as an apple shape. And so a person may have limbs that look pretty normal, so they don't look like a morbidly obese person, but all that obesity is right in the gut. It's central obesity. So that's what metabolic syndrome is. If you want to learn more about this study, not a lot more, but some more, there's a website run by the National Institutes of Health. And again, this is not the company's website. This is not somebody's blog. This is the website for clinical trials in the United States, and it's put there by the National Institutes of Health. You can go there, clinicaltrials.gov, and look at this. You can type in Protandim, and you'll see 3 studies listed. This is the newest one and one of them, and it tells you who's sponsoring it. You might note that one of the sponsors there is SomaLogic. It's another company. Their interest is in how to measure the proteins that your body produces. It's called proteomics instead of genomics. And they're using in this trial, their contribution is their new technology to measure more than 850 proteins in the plasma of these people, in the bloodstream of these people. And their hope is to generate a specific profile where they can diagnose metabolic syndrome just by a blood draw, not just metabolic syndrome, but they hope ultimately maybe hundreds of diseases by measuring these 850 different parameters. So that's an interesting coming together of technologies. And let me say it again, Bob Eckel, former president of the American Heart Association, chose Protandim to do this study. That's the only treatment being used. And if you put in Protandim, as I said, you'll turn up three such projects. One here is on non-alcoholic steatohepatitis, which means inflammatory liver disease. It's also a bit related to metabolic syndrome. And the other one is the use of Protandim to change lung glutathione levels in alcoholics who are oxidatively stressed because of the chronic alcoholic consumption. So all three of these are there. You should refer people to these. If you have a competitor, another product, and they want to take you on, ask them if they've got one clinical trial on clinicaltrials.gov, and ask them if they've got published peer-reviewed papers. And speaking of those, there's another one coming out in about a week. It's already available online. And that's the third thing I want to talk to you about. The dietary supplement Protandim decreases plasma osteopontin, that's a profibrotic factor, and improves markers of oxidative stress in muscular dystrophy MDX mice. Right? Duchenne muscular dystrophy is a human disease, and we have a mouse model of it that the origin is very close. The disease looks a bit different in mice than it does in humans. One of the reasons is as the muscles fail, a muscle in a mouse has very little stress load because a mouse weighs 20 grams and you weigh 70 kilos or more. If you have a metabolic syndrome, maybe a lot more than that. So this disease looks a bit different, but it's a very close biochemical model. And what was seen here, this was conducted, by the way, by my colleague Brian Singh when he was at the University of Colorado, Denver. As this study was in progress, about to end, he took a new job at Harvard University Mass General Hospital, and that's where the paper was written. So it's from Mass General and University of Colorado. One of the things we measured was T-bars. And again, every subsequent publication, every peer-reviewed study should hearken back to the first one and connect dots with studies in between. And this one does. You may recall in our human study published in 2006, Protandim decreased T-bars by 40% in humans who didn't have a particular disease. They were just getting older. It looked at young and old. These are pretty young mice, but they have a disease. They have this muscular dystrophy. Protandim decreased their plasma T-bars by 48%, okay, confirming that. Protandim decreased this profibrotic factor by 57%, right? This is not new either. This was done in the Virginia Commonwealth University study published last November, the one David mentioned in the journal Circulation. And virtually the same kind of result, Protandim in rats in that case and in mice in this case, reduced this profibrotic factor by 57%. Here's an enzyme I hadn't talked to you much about, but we actually measured it after that first human trial was published because we still had the samples. There's an important protective anti-atherosclerosis enzyme in your plasma, abbreviated PON1. And it's associated with the good form of cholesterol, and it prevents the oxidation of the bad form of cholesterol, which promotes plaque buildup in the arteries. And here is an example. This is not by gene regulation. It's actually by preventing the inactivation of PON1 by lipid peroxidation products related to T-bars. So T-bars is a bad thing. One of the bad things it can do is inactivate good essential enzymes. So the PON1 in these untreated animals is lower than it should be. When they're treated with Protandim, that inactivation is prevented, the PON1 levels rise, and now you have the adequate amount of protection by PON1. Is PON1 important? This is a paper that says low paraoxinase, PON1, predicts cardiovascular events in a study conducted of patients at risk for heart disease. So yes, and I've shown you a similar paper before showing that T-bars elevation also predicts cardiovascular events. This is a compilation of measurements of PON1 and a number of diseases, and I'm not going to read them all to you, but you can see the first one after the normals, which has a level of normalized here of 100%, is the metabolic syndrome, and followed by diseases like autism, type 2 diabetes, a whole bunch of things you can read there. And at the very opposite end of the spectrum is the normal people in our first human study who were treated with Protandim. And you can see that their PON1 is significantly elevated above the normal level and way above the level seen in these diseases. And then finally, I want to show you a very interesting measure, and I think you can see that okay. These are magnetic resonance images, an MRI, of the calf muscle, the lower leg, of these mice that have this muscular dystrophy. And the upper two panels show two of these MDX mice that were treated with Protandim. The bottom two panels show MDX mice that were not treated. And I don't expect you to be able to read MRIs necessarily, but I want to point out what's different here. You can see there are white areas, and these are probably areas of fibrosis within this muscle called the gastrocnemius muscle. It's your calf muscle, and the corresponding muscle in these animals. And in the upper level, in the upper Protandim-treated animals, you can see much less of that. And if the computer can measure the areas of white, quantify it, and what you see here is there was a 38% decrease in the Protandim-treated animals of this so-called anomalous MRI area. This part of the study actually fell just short of statistical significance only because there weren't enough surviving control animals to bring that number down. There were only 10. And doing MRI on a mouse is fairly difficult. He has to be anesthetized. And often the ones that died during that were because they died during the anesthesia. It's a little more difficult to give anesthesia to a mouse than to a human. But in summary, this study confirmed things you already know. Protandim decreases T-bars. Protandim decreased osteopontin, a pro-fibrotic marker. Protandim increased this new enzyme I'm talking about, paraoxidase 1, PON1, a protective anti-atherosclerosis enzyme. And Protandim decreased the anomalous MRI signals in the leg muscles of these animals. So thank you very much for your attention, and I hope you learned something.